Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Right. Good. Appreciate being here, and especially being invited by Ernest. I'll assure you he's doing a splendid job, so no jokes. Jokes would have been fine. So the topic of what I'll present to you today is what we're after conceptually in terms of our laboratory. Maybe you can see these are not, and that is, how do we intervene in a predictive manner in the types of complex pathologies that we might be interested in? So, heard a very interesting example just previous. This, I'll give you the exact disease that we're focusing on in a minute. But in general, these are inflammatory diseases in which you see many members of the immune system, innate and adaptive immune system, cytokines, then affecting also what's going on in tissue with tissue cells and blood vessel cells and so forth to lead to a pathology. Because of course what you'd like to be able to do is to say, can we figure out where the best targets would be? They're likely to be combinatorial. Can we predict what would happen if we intervene? So what kind of combination of experimental measurements and computational analysis can we do to tell us how to do this? So this particular application, some of you may be familiar with, some of you not. It's a disease that's not very well studied at all. There's something like only a handful of NIH R01s on this disease, despite the fact that it affects 10% of all reproductive aged women from puberty to menopause, and that's a very, very, very large number of people. It's a major association with infertility. Almost half of women infertility is associated with this disease. The economic burden is estimated to be similar to that of rheumatoid arthritis. So if you think of all the work going on in the world on rheumatoid arthritis, this is roughly a similar disease in terms of debilitation and prevalence and so forth. Nonetheless, very little is known about it. So what actually is known? Well, it's a disease in the peritoneal cavity in women that tissue spreads to places that it's not supposed to be and invades and essentially becomes tumor-like with respect to then causing pain, debilitation, and some organ dysfunction. Schematically, it looks something like this, that reflux tissue during menstruation can end up in the peritoneal fluid, and it ought to be cleared by the immune response with no problem. These cells ought to just die and go away, and no problems at all. Nonetheless, what happens is they persist. They're not cleared by the immune response, and in fact, they invade into the tissue, and they become these proliferative lesions that you see on the bottom right over here, down in the tissues where they actually form little glandular structures, and as I said, they can lead to angiogenesis. They can lead to neuronal issues and generally to pain. So it's an invasive disease of some tissue, some cells that come out of one tissue and move into another tissue, and instead of being cleared by the immune response, they are allowed to survive, proliferate, and invade. So many, many similarities to cancer in terms of being an invasive disease of essentially epithelial cells. Now, it is appreciated to be a disease of dysregulated immune system behavior, and I won't go through all the arguments. I'll just give you a recent review article here. But many aspects of the immune system seem to be dysregulated from the studies that have been done, altered activities of the different types of T cells, natural killer cells, possibly autoantibody production, generally increased inflammation, some hyperactivation of some inflammatory pathways. The bottom line is the premise seems to be that there's something dysregulated in the immune system, very unclear, that dysregulates then these tissue cells that have come out of the uterus and are found in then landing on various tissues, and the dysregulation of the immune system allows them to survive, proliferate, and invade. So it's a fairly vague hypothesis, but that's really the extent of it. So what we're after is, with clinical patients and some basic cell biology studies, something like the following: a multi-scale, it's got to be integrated clinical experimental data and computational analysis to deal with all the moving parts. And somewhere in here lurking is some formal multi-scale, multi-component analysis. That's not the really way we've really approached it yet. We've looked at it in a number of different pieces and tried to sew the pieces together mathematically with what you might call a hybrid approach. Nonetheless, in essence, it's a multi-scale problem. So what are the scales? Well, one of them is in the peritoneal fluid and proximal to the tissue, you have immune cell communication networks. All the different type of immune cells are communicating with each other, producing different cytokines and chemokines, changing the numbers of types and the differentiated state of the different types of immune systems there. So part of it is to figure out what's dysregulated in what cells are there and how they're communicating with each other and telling each other what to do. Of course, all that communication in any given cell type is governed by the intracellular signaling pathways. So inside of each one of these types of cells, there's multiple pathways governing what cytokines are produced and what's going to happen next. You might wonder which cell type to pick. Should I be studying this in all these different cell types? And maybe so. That's part of the question. Now, they're giving these things, so there's something going on with the signaling networks that are changing the cytokines and chemokines they're producing that affect each other. But in the end, the disease happens in the tissue. The disease is of these ectopic endometrial cells that survive and proliferate and invade tissue that they ought not. So you also have to study what's happening down in the tissue level with the endometrial cells and how are they talking to each other and other cells in the tissues. Well, it's by proteases and growth factors that they're trading among one another, essentially analogously to the cytokines and chemokines in the immune system. And of course, this is governed then by the signaling pathways inside these tissue endometrial cells. So you see the different layers. We have different cell types, immune system network, tissue cell networks, the pathways inside of each one, and somehow we need to sew this all together. And in the end, we want to tie these two arms together, that what the immune cell networks are doing influences what the endometrial cell networks are doing, and that is actually where the pathology resides. And someplace in this, then, where would you intervene? Where would you add a drug? Would you add an antibody against one of the cytokines? Would you have a small molecule signaling pathway inhibitor? If so, which cell type, which pathway? So that's the idea of that. It's this multi-scale and multicellular type. So let me run through these. And what we've done is try to analyze each one of these different layers and sew them together. So let's start with the easiest, and this is where our study began and where some others in the literature have also landed, but really in a much more traditional way. So let's look at the immune cell cytokine communication networks. And one can do things like this with patients. And we've done this with patients in right here in the Boston area so far with Keith Isaacson and Newton Wellesley Hospital. We've got collaborations in Norway, collaborations in Sao Paulo. And an interesting question there is, yes, are there geographical types of stratifications there? And we're just learning this. This is the Boston area study. And so one can have, in our case, it ends up to be 50 or 60 patients who have undergone laparoscopy, which is really the only diagnostic for whether you have this disease or not. A surgeon has to actually go in and look. And you can look at the different patients, and there's some that don't have the disease. In a noticeable way, they can't find any lesions. Some seem to have major lesions, fewer lesions. Some might be on hormone therapy. And the first thing to measure is what's actually in there in the peritoneal fluids. What are the immune cells producing that's influencing each other? And so you can measure that in a fairly high throughput way by Luminex. So measuring about, in our case, a panel of about 50 different cytokines and chemokines. And what one sees, what's plotted here, is across the 4 different stages: controls, mild disease, severe disease on treatment. You can look one by one and see that a large number, a couple dozen of the cytokines and chemokines, are overexpressed or underexpressed in one of the conditions versus another. And that's sort of the state of the literature right now, is people argue over which one of these is the most important. because they might potentially be a drug candidate. So where we tried to go beyond this in a multivariate way was just to start with an unsupervised analysis, a non-negative matrix factorization of these molecular data. And we did it independent of the presumed disease status, because what's very interesting is the disease status, by just counting the number of lesions, really does not relate very well to actually symptoms, pain, debilitation, and so forth. So if you're actually a patient, you care more about your pain and your debilitation than do I have a lot of lesions or fewer lesions. So we did the unsupervised analysis independent of the actual pathological histological assessment, and we could find two interesting clusters. And then if you go back in an unsupervised way, it turns out that this cluster with the more severe sets of cytokines upregulated are substantially enriched in the severe pain patients, even though they didn't necessarily cluster with, as I said, the number of lesions or even being on treatment. Okay, from this you could identify what actually is in the signature. There's a set of about 13 of these cytokines and chemokines, just depicted as CKs here, that form a signature of this pain-associated cluster of immune system network communication molecules associated with the pain patients. Although, again, we didn't classify them that way. That's just the association as it turns out. Now, to try to figure out what's going on with the cell types, you'd like to do this by flow cytometry. It's highly variable, the types of cell samples right now one can get from the peritoneal fluid, so that really wasn't an option, at least at the medium presence. So we went into the databases of expression in the immune system, of which there's many. And so what you can find is, for instance, cytokine expression across many different cell types, cell lineages, and activation states, and what their cytokine expression profiles are. And now you can map that quantitatively against the signature that we found enriched with our most diseased patients, and calculate what cell types are to be greater or lesser represented in terms of the cytokines and chemokines they're producing. In terms of the receiving end, you can do similarly for cytokine expression. Say what cytokine receptor expression for the various different cell types, should they be overrepresented or underrepresented with respect to being able to bind the cytokines and chemokines that are found in that signature. Putting these together, you get an inferred communication network that says what cell types are talking to each other, which cytokines are involved in one cell type talking to another versus other cell types talking to others. Who's the producers, who's the receivers, what are the cassettes of cytokines. And it leads to this, and one of the early hypotheses that comes out of this is that this is a activated monocyte-driven disease, that people could choose any immune cell type they wanted. Some people are focusing on CD4s and CD8s and NKs and so forth, and this inference model pulls out activated macrophages as actually the central driver. We go back into the patients and now look at their macrophages, pull them out of the peritoneal fluid, measure what they're making, and what they're making, in fact, is highly enriched in the patients in the cytokines that were in the signature. So we could find in the patients versus the controls enrichment of macrophages producing the signature largely that we said the inference model said that they're making. Okay, so that's the immune cell communication network. Who's driving it, and what cytokines and chemokines are they making and sending to each other? And of course, you might want to play through all of these inferences and block one cytokine or a chemokine here or another and see if you can break these loops. And that's something, of course, one can imagine doing, not so much in human patients. Unfortunately, there's not very good animal models for this disease, so one really has to think about how one can put this together. Now you say, okay, we're going to focus on these activated macrophages. What's going on inside of them that's causing them to be activated in this particular way? It's not even a classical polarization. If you know inflammation, it's not precisely M1 or M2. There's a mixture of cytokines that they're making. So how you would actually categorize these macrophages isn't clear. So you can now go into the macrophage gene expression databases and see what gene expression co-enriches with the activation signature of the cytokines that they're making. And and with some inspiration from Ernest Frankel, we focused on transcription factor activities associated with the enriched co-expressed genes, the other genes that were co-expressed in enriched ways with those key cytokines, and found two most germane upstream signaling pathways. One was IKK, that's upstream of NF-kappa B. That was entirely expected. It's just a highly activated inflammatory signature in many cells. The Jun kinase pathway was also co-identified, and that was entirely novel. That's not really been appreciated to be a driver in a lot of these inflammatory diseases, and especially not in endometriosis. So we wanted to test this specifically. So from this enrichment study, we actually could rank order different kinase pathways according to the strength in which they ought to be contributing to the production of these cytokines and chemokines in the enriched signature. And we could show that you actually get the right rank order, that on the right-hand side, if you come in with an inhibitor of Jun kinase, the blue squares show that you have the strongest diminution of all the cytokines and chemokines that were in that signature. The next greatest prediction was the IKK pathways I showed you. That's the next block over, the lighter green, still a fair amount of blue and a little bit of red. And then as you go farther over, some other pathways that people had previously presumed, especially P38 and JAK, as big drivers of activated macrophages. You see much more red, much less blue. There's much less effective inhibitors on those. So we validated that the Jun kinase pathway was the most strongly associated upstream signaling pathway for producing the activated macrophage cytokine chemokine signature. And I'll note there's an unnamed pharma company that has especially selective Jun kinase inhibitors that they are starting clinical trials with. Okay, so that's half. What's the other half? How is this inflammatory dysregulation linked to the actors, the endometrial cells, that epithelial cells that actually have to invade and form these tumors? Okay, we've identified what's happening out in the immune system that's dysregulated. Now, how is this affecting the actual endometrial cells in their lesion formation? Well, so now we have to study what's driving them molecularly, what's driving these cells to proliferate and invade into the tissue, and what are the intracellular pathways that might be most strongly associated with that, because there again might be targets now on the endometrial cell end rather than on the macrophage end. So we'll look at both these together. So there is some evidence that exists previously. Some people have done small-scale in vivo studies of endometriosis to see what kind of receptors are most strongly activated. Interestingly, they came out with the ERB family, the epidermal growth receptor family, which is interesting because that's a big driver of many cancers, especially hormonal-related cancers, things like breast cancers. And there's a lot of similarities between the properties of these invasive endometrial cells and invasive breast cancer cells that we're exploring. So that allowed us to narrow in on this ERB family, epidermal growth factor receptor family, for what might be dysregulated in the endometrial cells. But an interesting question is, where do those growth factors actually come from? They don't come from the immune cells in the perineal fluid. They're actually produced by the cells themselves in a very interesting manner, in which the cells themselves make their own growth factors. So they're put in the cell surface, but they're there in a very unusable manner until proteases activate them, and they become soluble, and they can come back and bind to the EGF receptor and other ERB receptors on the same cells and on neighboring cells. So what appears to be the case is there's something in the dysregulated inflammatory system that's driving the activities of these proteases that then make more EGF receptor ligands that turn on these cells and cause them to invade. That's an interesting hypothesis. So we carried out an experimental cell biology study on this, taking some of these invasive endometrial epithelial cells and measuring in them their protease activities at the cell surface, what growth factors they're shedding into the environment, what their intracellular signaling pathways are that are downstream of that, and might be governing the migration. So we measure all these things simultaneously: the kinase signaling pathways inside, the cell migration, the protease activities, the growth factors that are shed into the environment. If we put all that together, and the data just looks something like this, the intracellular phosphoprotein signaling across multiple pathways and multiple treatment conditions that one might see in the peritoneal tissues, the protease activities, various different proteases under all these conditions, the actual growth factors and receptors that are shed proteolytically into the peritoneal fluid, and then their migration behavior. And by a Bayesian network pulled all this together to say who's driving what. So downstream of the cytokine and growth factor stimulation that would be in the peritoneal fluid, what pathways are being strongly driven. Interestingly, here's our friend Jun kinase again, actually in the endometrial cells themselves, which might give you a double strong therapeutic effect. Other downstream phosphosignaling pathways in which you see these arrows in the Bayesian network are crosstalk, influencing the activity of these things called atoms. Those are proteases. Those proteases are what release the growth factors that identifies what the key growth factors are that are released that then drive motility. So what this Bayesian network model allows us to do, and I won't go into the details, it allows us to predict where the best drug combination therapies might be. If you want to inhibit motility downstream of pretty much any type of stimulatory environment, because you don't really know the stimulatory environment, what two of these things in combination would you use to inhibit that most strongly? And it turns out we could predict that very nicely, and it turns out it's an inhibitor of one of these receptors in which its ligand has feedback, and then one of the downstream kinase pathways that governs most strongly the protease activities that govern the ligand release. And we could test that in the cells using different individual inhibitors and different combinations of inhibitors and show that we can really in fact prevent this invasion in the strongest way with the combination predicted here. All right, so we know what's going on in the immune system cells. We know what's going on in the endometrial cells. Now how do they talk to each other? Is there any information here? Can it be analyzed for how they connect? Well, so what you want to do is concomitantly in the same patients in which one has measured all those cytokines and chemokines in the immune system, you'd also like to then measure all these protease activities and the growth factors and receptors that are shed in the same patients. And fortunately, we're able to do that, having kept the samples. And now we're trying to see, are there relationships between the molecular drivers in the immune system dysregulation and the molecular drivers in the endometrial cell invasion? So pulling these data sets together, now this is just a simple topological correlation network model. It needs to be trimmed down a lot. This is a very preliminary model. But what one can see here is some combination of, in yellow, some of the cytokines and chemokines that are in the peritoneal fluid, some of the growth factors that are shed from the surface, some of the receptors, some of the proteases. And if you look at the things in the peritoneal fluid in the immune system dysregulation that are most strongly upregulated or downregulated, you can see them represented here as central drivers of, at least associated with, correlated with, the growth factors that were driving the endometrial cell invasion. So this is a very first preliminary step toward tying the two together. The key things that have been dysregulated here, how are they associated with the key things that are dysregulated there? So where we are in the end is I think we've learned a lot just from pulling a number of different data types together, mainly from clinical patients, but also in combination with cell biology, biochemistry studies, and some databases that are available on gene expression. So we've learned something about what's dysregulated among multiple cell types in the immune system, what one of the key immune system cell actors appears to be, what some of his or her key pathways appear to be intracellularly that lead to the dysregulated signature, prediction of what pathways to block to reduce that, validate that. On the side of the endometrial cells, what growth factors are driving them, what pathways might we block there, that regardless of what's in the immune system, might block the motility introduced by these autocrine EGF receptor ligand interactions, and then how are these immune system cytokine network dysregulation signatures associated with what's dysregulated in the endometrial cells. So in some places there's mechanism, in some places there's purely correlation. And I think you can imagine ways to try to put this into a nicely fused, integrated multi-scale model. We just haven't done that yet. This is hybrid, as I say. So thank you especially to Keith Isaacson and Linda Griffith, who have led the clinical side of the study, and Michael Bestie, Miles Miller, Aaron Meyer, and Abby Hill. Thank you.
Any questions? Please repeat the question.
Okay. Yep, start here.
So that when you build the mathematical model at the end, how do you identify the parameters? Do you do cell line measurements of growth and death rate? Or how do you build these models?
Okay. The question is, when we build these mathematical models, where do we actually get parameters for growth rates, death rates, and things like that? In most of these are not really parameterized models. Okay, there's no rate constant per se. Right? In Bayesian networks, you just have conditional probability distributions. In these, you just have probabilities of correlation. Now, when we did this, we actually had numbers for things like motility and the signaling and the proteases and things like that. So at this point, these aren't really parameterized models in the sense of a priori predictions in that sense. We can compute through these, but there's no specific parameters assigned with rates of mechanistic processes. That would be a future thing once we had confidence that we've got the right relationships and associations here that I think we need to work harder to convince ourselves of. You had?
Yeah, so actually my question was about the Bayesian network inference. Typically, how many variables do you include and how many independent samples do you have?
Yeah, in this, the independent samples came from, this was a cell biology study. So we drove it with about 8 different cytokines and chemokines, and I think we had triplicates of all the conditions. Then you see all the components that are in there. So what this is not a thousands and thousands and thousands of data point type of analysis. That's something we've learned with a previous student in the lab is how efficient one can be with data to generate these kind of networks, and you can be more efficient than sometimes people think you can. It's more the quality of data than the number of data points you have in terms of covering dynamic ranges and signal to noise ratios.
Time for one last question if anyone has one. All right, if not, let's thank Doug again.